Hello my friends, in this video I want to show you the autosomal DNA predicted phenotype traits and GD match results of Upper Paleolithic Siberian Malta boy, also known as MA1. I've already done a video on Afontova Gera 3, make sure you watch that. First, I would really appreciate it if you subscribe to my channel and left a like. Trust me when I say this, this was a difficult task to accomplish. I had to literally assemble this genome part by part, each of the 4 parts weighing over 5 gigabytes in unmapped format. I do a lot of work and it seems to go mostly unappreciated here on YouTube. In case you aren't familiar with Malta boy, he is a part of an Upper Paleolithic population known as Ancient North Eurasians. And Ancient North Eurasians contributed to the ethnogenesis of Europeans, peoples of the Middle East and South Asia, and of course to Native Americans. There are even traces of Ancient North Eurasian DNA in Northern Africa and Mongolia, although non-Mongol East Asians such as Chinese, Japanese, Koreans, Vietnamese, they don't really have any ancestry from this group. It is debated whether or not the Ancient North Eurasians were the first humans to domesticate dogs. However, despite this debate, it is known for a fact that every domestic dog today traces their ancestry to the dogs domesticated by this human group. We know this from analysis of dog haplogroups. Regarding this sample this video is about, Malta boy lived 220 centuries or 22 millennia before the Common Era. His remains were found in Irkutskaya Oblast, located in southern Siberia. Malta boy was found buried together with a figurine made out of mammoth bone. Such figurines are called Venus figurines. Plenty have also been found in Europe. Carved from ivory tusk of a mammoth, these images were typically highly stylized and often involved embellished and disproportionate characteristics of female body parts, typically the breasts. It is widely believed that these emphasized features were meant to be symbols of fertility. Around 30 female statuettes of varying shapes have been found in Malta. The wide variety of forms, combined with the realism of the sculptures and the lack of repetitiveness in detail, are definite signs of developed, albeit early, art. This boy had haplogroup R star, which derives from R, but doesn't fall into either R1 or R2 subclade. There are no modern humans today with haplogroup R star. This boy's paternal lineage seemingly disappeared. His mitochondrial DNA is U, which is nowadays a lineage most common in Europeans. This is what he is predicted to look like. With my NOSHACOT, he's got dark brown color eyes, Greek-shaped nose, and black hair. I was actually surprised that he scored 90% likelihood of Greek-shaped nose. I was expecting an Ancient North Eurasian Malta boy to score maybe snub-shaped nose instead, but I guess preconceived notions do exist to be broken when faced with reality. With Y-sec, he's also predicted to be quite dark. When it comes to his hair shape, my hair shape predictor tool predicted for him wavy hair texture. Although this was only done on the basis of 8 SNPs, my eye shape predictor tool predicted him to have Estonian, followed by South Asian, followed by Amerindian eye shape, so kind of, not East Asian eye shape, not epicanthic folds. He does not have any derived variants in EDAR either. And he most likely has blue eye haplotype 1 based on one genotype in the OCA2 region. And he does not have BH2 or BH3 or BH4, and that we know for a fact. Does not have blue eye haplotype 2 or 3 or 4. So pretty dark in terms of color of eyes, hair. And when it comes to skin tone, he's probably got light brown or olive skin tone. He does have some light color variants that I highlighted in blue on the screen in SLC45A2, KITL-G, ASIP, and IRF4. However, despite this, he is missing some very important variations for light skin, which you cannot really have light skin without having these genotypes. So he probably has light brown or olive skin tone. And when it comes to Snipper-Free actually predicted him to have white skin, which I found pretty interesting because Snipper-Free uses completely different SNPs for the skin color prediction. And I don't know how reliable they are because I haven't never done a study on skin tone, but it is what it is. With Snipper-Free, he's got white skin and he's got black hair and brown eyes with Snipper-Free. It seems that all the 3 tools, NOSHACOT, Y-sec, and Snipper-Free, agree to the fact that he's got black hair and brown eyes. At least that's a plus. This is what he scores with the Eurasian 36 on GD match. I separated the parts, because the Amerindian and Volga-Ural have the same color, for example. I had to separate where Amerindian ends and Volga-Ural starts. Same goes for Finno-Scandian and South Asian. They have the same color, unfortunately, so I had to draw a little line on the screen to show you where one ends and the other one begins. This is what he scores with Eurasian K13. He is scoring 8.3% West Asian, which is pretty typical for ancient North Eurasians in general, and also 22% South Asian. These people have very strong affinities to South Asia and West Asia, and a lot of people don't understand this tie that exists between ancient North Eurasians and populations of South Central Asia, such as like Pashtuns, Baloch people. There is a lot of continuity, and a lot of the drift that's specific to Baloch people was present in ancient North Eurasians, such as Malta Boy or Afontova-Garatri. Less so in Afontova-Garatri, more so in individuals like Malta Boy. So this is what he scores with MDLPK23B. Ancestral Altaic here is actually a component that's meant to represent ancient North Eurasian specific drift, so that's why he's scoring so much of it. And with the Oracle, he's basically getting modeled as a mixture of like Siberian plus Siberian. Tubalars are a group of people in southern Siberia. Khans are a group of people in like northwest of Siberia, bordering on the Ural Mountains. And this is what he scores. Now, this is what he scores with MDLPK11 Modern. As you can see, he's scoring a lot of Iran Mesolithic and EHG, and EHG here actually represents Caucasus-specific drift. So he's got a lot of Caucasus admixture. If you look at this and you just take it as a result of a modern individual, this is a modern individual with a lot of Caucasus admixture, right? However, with the Oracle, we see that this is a typical result for an Upper Paleolithic Siberian. It matches the data that's in the Oracle, right? So that's what people miss all the time, is that ancient North Eurasians actually had a lot of affinities to like Pashtuns and Baloch and all these South Central Asian groups. This is what he scores with Harappa World. Once again, you can see here 10% South Indian plus 23.5% Baloch. There's a lot of affinities between this individual, this ancient North Eurasian, and modern South Central Asians who score a lot of Baloch and South Indian with this calculator. There's also affinities to American Indians, to Native Americans, but there is stronger ties to South Central Asia than even to Native Americans here. With the Oracle, you see it's Jats plus Finns, or various North Indian groups plus Finnish people. This is what he scores with PanDNA LK12 Ancient. Once again, you see that he's scoring 20% Caucasus Hunter-Gatherer. If this individual did not have any Caucasus affinities, and he was just like a mixture of Western Hunter-Gatherer plus some kind of Tian Yuan, Western WHG plus Tian Yuan would not score 20% Caucasus Hunter-Gatherer. It would score 34.5% European Hunter-Gatherer, and the rest South Asian and Amerindian and something else. Something that's not Caucasus Hunter-Gatherer. So these ancient North Eurasians, and this is not just the result of Malta 1, this is the result of Afontova-Garat 3 and 2, although Afontova-Garat 3 to a lesser extent, they're a little bit more northern, a little bit less affinities to South Central Asian groups. But Malta 1, and you can see with the Oracle here, it pretty much exactly matches the reference population for Malta 1. Malta 1 has a lot of affinities to these North Indian and Pakistani groups, and with the Oracle, he's actually getting modeled as a mixture of basically entirely Malta 1 plus a little bit of hunter-gatherer admixture, European hunter-gatherer admixture. It's a pretty close fit. I actually really like it. I don't see a lot of results that have such a close fit. This is what he scores with PanDNA LK10, and you can see once again 26% CHG here, a lot of Caucasus hunter-gatherer affinities. That doesn't mean he has Caucasus hunter-gatherer. He can't have Caucasus hunter-gatherer admixture. Caucasus hunter-gatherers were not a thing at the time that this individual lived. He's a Upper Paleolithic individual. Caucasus hunter-gatherers are a Mesolithic group, but he is nonetheless scoring 26% CHG with PanDNA LK10. And with the Oracle, he's getting modeled as a mixture of Mardvin plus Caritiana or Bolivian. Basically, it seems like 3/4 Northeast European plus 1/4 American Indian. That's what it looks like. There is also Belarusian and Finnish plus Caritiana. There is also Chuvash plus Bolivian and Chuvash plus Mayan. And this is what he scores with Ancient Eurasia K6. A lot of ancestral North Eurasian here. He's not actually scoring any Natufian, which is kind of interesting. That's the only category that he's not scoring anything from. He is even scoring Sub-Saharan African, which, actually you would expect an Upper Paleolithic individual to score more Sub-Saharan African. So it's surprising that he doesn't really have any Sub-Saharan African admixture with any of these calculators. But he is getting modeled as a mixture of Malta 1 plus Goyet, or basically Malta 1 plus Epigravetian with the Oracle. And finally, we're moving on to his results with Gedrosia K3. He's scoring mostly West Eurasian, but there is a little bit of East Eurasian and Sub-Saharan African. Well, there is no Sub-Saharan African admixture. There's just affinities to Sub-Saharan Africans that exist because this individual lived in a time before a lot of the West Eurasian drift happened. And with the oracle for this calculator, which it doesn't work on GD match, unfortunately, I had to make my own oracle. You know, basically look at the spreadsheet, put that on CSV file format, and make my own oracle. With the oracle for this calculator, he is closest to Tajiks and various South Central Asians. Now we're going to take a look at his traits, his results with Nebula. After I show you his results with Nebula, I'm actually going to move on to show you his results with my trait predictor as well, but we're going to start with Nebula. So don't get discouraged and think I'm not going to show you that. I'm going to show you both, okay? But let's show you his Nebula results. So for ADHD, and this study covered 27 genetic variants, which is, I guess it's decent coverage. We're going to have to look at the p-values to see how statistically significant it is. And his 91st percentile, very high genetic predisposition to ADHD. 91st percentile for ADHD is no joke. ADHD is super common, so being top 9% for that is, it says a lot. So let's see how important these variants are. Okay, so the variants here actually don't have much of an impact. You see a p-val like this, it's a pretty high p-val. Let's see how big the study was. Okay, so the study was on 225,000 people. For a study that's this big, a p-value like this, even though it's the most significant on the list, it's actually not very significant at all. So the ADHD result may be not very representative of reality. For bipolar disorder, and this study focused on 64 genetic variants, pretty good coverage once again, and he's scoring 34th percentile, below average genetic predisposition to bipolar disorder. And let's see. Okay, so the variants here, this is a decent p-val, but it depends on how big the study is. Let's see how big the study is. How big is the study? Does it say? Does it even say here? No, it doesn't mention. Okay, so I guess we're gonna have to leave this. If you want to research into this study, Mullins 2021, please do it. I don't really care about it too much, but let's scroll past this, and this is what he scores for autism spectrum disorder. 61st percentile, which means above average genetic predisposition to autism spectrum disorder. And let's see the S&Ps. Okay, this is not very significant. None of this is really significant. We can move past this. For type 2 diabetes, and this is crazy, by the way. I was shocked because I saw this before I'm making this video. I saw all of them, but I do need to prepare a little bit before I start talking about this stuff. So for type 2 diabetes, his 99th percentile, very high genetic predisposition to type 2 diabetes. By the way, something like 1/3 of Americans has type 2 diabetes. So if you're top 1% for a trait where 1/3 of people have it, your odds of having it are astronomical. You definitely have type 2 diabetes. If you live in America and you have the kind of diet that Americans, consume, you're definitely going to develop type 2 diabetes if you have this kind of a genotype. But I think maybe in the environment that this individual lived in, like in Paleolithic Siberia, where they hunted mammoths and were always on the run, they were always doing exercise, they weren't getting much sugar, maybe in this kind of environment it wouldn't matter all that much. Or maybe alternatively we can turn on the analytical scientist brain right now, and we can say that probably there's a chance he has such a high risk score for type 2 diabetes because for some reason his people evolved to, I don't know, be more responsive to sugar because there's a lack of sugar in their diet. Just throwing that out there. That might be the case, but I don't know. I haven't seen any other ancient North Eurasian results with Nebula so far. So this is based on 568 genomic variations, 568 S&Ps. That's no joke, guys. We're gonna have to scroll through a couple pages of these results, and actually let's see how significant they are. Very significant. So this is incredibly significant. This is also incredibly significant for the score. This is also, this is all really significant stuff. I don't expect to see such low p-values for a trait like type 2 diabetes. I thought it was very based on environment, not so much in genetics, but maybe I was wrong. Let's see how many people that is. Okay, so a lot of people. That explains it. When you have this many people in the study, you're going to end up having lower p-values. Still though, it does say a lot. Like p-value like this, it's no joke. Let's scroll past all of these pages. By the way, all of these pages are his genotypes for type 2 diabetes. We're going to scroll past all of them. There's a lot. There's a lot that goes into this calculation, guys. We're going to scroll past all of these results. And now for cerebral cortex surface area. This basically has to do with your cerebral cortex, how you remember stuff. From my understanding, it was a while when a lot of time passed before now and when I took biology in high school, so I don't really remember what like cerebral cortex does, but he's got 92nd percentile, very high genetic predisposition to larger cortical surface area. And once again, this is a pretty important, pretty significant result here. So how big is the study though? How big is okay, 50,000. I guess it's not as impressive as I thought. We're gonna scroll past all of this. This is continuation of that. We're gonna scroll past all of this. Wow. And finally for height. So for height, he's actually 27th percentile, below average genetic predisposition to taller stature. I don't know. I was under the impression that ancient North Eurasians would be tall people. I don't know why, but I guess it would make sense for them to be kind of short, because like I don't want to come off as racist, but like Hispanic people tend to be short, and Hispanic people have, especially the Hispanic people that have more of the Native American genome, more of the Native American DNA in them, less of the Spanish, more of the Native American. So for this result, this is, by the way, this is based on 697 genetic variants, so this is also a very legit result. And the biggest contributor here is this and this. Once again, very significant p-val. But let's see the study size. Okay, once again, big study size, which means you're gonna end up having lower p-vals. This is less impressive. Seeing a very low p-val is less impressive when you see that the study size is 250,000, right? Not as impressive when you see that. So we're gonna scroll past all of this height-related stuff. We're gonna scroll past all of this, and we're gonna move on to what comes next. Wow, there's a lot of height. Okay, male pattern baldness. For male pattern baldness, Malta boy has got 4th percentile, very low genetic predisposition to male pattern baldness. He's not gonna go bald, and this is based on 71 genetic variations. How many? What's the sample size? I don't see any sample size here. So 71 genetic variations, 4th percentile, definitely doesn't go bald. And okay, so this is the biggest. This is an incredibly significant p-val. I don't think the p-val can be closer to 0 than this. How would you calculate it? I mean, and he's got AA genotype here. By the way, the AA variant is the frequency of the A variant is 15%, which means the frequency of the AA genotype would be 0.15 times 0.15, which is like 2.5%. So basically 2.5% of people have AA genotype like this individual, which would protect them from male pattern baldness. So this is the biggest contributor to his results, and this is why he's scoring like this with this calculator. We're gonna scroll past all of this, and finally we arrive to schizophrenia. This was done with 53 SNPs, and his result is 68th percentile for schizophrenia, above average genetic predisposition, but nothing too alarming. We're gonna scroll past this. We don't really care about that. And finally, birth weight. Well, not finally. I have a couple more For birth weight, he's 99th percentile, very high genetic predisposition to heavier birth weight. Definitely weighs a lot as a baby. Myself, when I was born, I weighed like 5 kilograms. So, ancient of Eurasian genes strong, I guess. So 99th percentile, very heavy, very high genetic predisposition to heavier birth weight. And let's see, Alzheimer's disease. Okay, so for Alzheimer's disease, 99%. Okay, this is kind of sus, right? Oh, so he's got GG here. Right, and this is why the score is like this. This is why it's 99 percentile, because he's got the genotype right here, and it's very rare, and it leads to a increase in the risk of Alzheimer's. And you see the G allele is found at frequency of 14%, 0.14. So for the GG genotype, which is his genotype, you have to multiply 0.14 by 0.14, which, I mean, it would be a bit over 1% in terms of the it would be 2% actually. So 2% of people have GG genotype like this individual, which greatly increases the odds of Alzheimer's. And this is why his score is like this, incredible score. Now, oh, that's actually it. Now we'll be taking a look at his results with my genome analyzer tool, which is on my website and on my GitHub. So let's begin. He has GG in Comtes-Vall methylation, meaning Vall genotype, meaning warrior genotype, basically quicker reuptake of dopamine, less dopamine in the system. And he's got GG in DRD2 spirophrenesin pro variation, which means higher number of dopamine D2 receptor sites in the brain and higher risk of schizophrenia and various other mental health conditions that involve an overabundance of dopamine. But I think this is sort of counteracted by this genotype right here. He's got AA genotype in TAC1. The A allele here is like the reference name for it is A1, and for the G allele, the reference for that is A2. So he's got A1 genotype in TAC1. When I say A1 or A2 genotype, that's basically what I mean. A1 corresponds to A allele and A2 corresponds to the G allele. So he's got A1 genotype in TAC2, in TAC1, which is super rare. Well, I mean, it's not like impossible to find in humans, but it's just kind of rare, and it leads to a significant decrease in the number of dopamine D2 receptor sites in the brain. It's basically like taking like Zyprexa. It's like you're taking Zyprexa and but you're not taking any drugs, you just live like that. You just have much lower availability of dopamine D2 receptors. And it does lead to increased likelihood of alcoholism, Parkinson's, ADHD, and various other illnesses. What's interesting about this specific variation and the distribution of the allele frequencies here is that in Neanderthals and various monkeys such as gorillas, chimpanzees, orangutans, all the other apes, as well as monkeys like baboons and macaca, they all have AA genotype. They all have the A1 genotype in TAC1, just like this Malta boy, kind of a monkey genotype to have in a way. So he's got CT here in 5-HTTLPR. He's got short form 5-HTTLPR. Just like most of you guys, he's got a slightly higher odds of depression because of that. Okay, doesn't have long form 5-HTTLPR, I guess that's all we can say here. Does not carry the European lactose persistence mutation. And heterozygous for OXTR, for the main variation OXTR, which means one sociopath variant for reduced OXTR expression and lack of empathy, together with one variant for higher levels of empathy. It's the same genotype as me. Actually, I have TT here as well. So overall, these two genotypes are exactly the same as me. I wouldn't say I'm a very sociopathic person, so I guess this guy probably wasn't a sociopath either. He's got CC genotype here, which leads to 7-fold decrease in the risk of type 1 diabetes. Does not have type 1 diabetes. By the way, don't confuse type 1 and type 2 diabetes. Type 1 diabetes has to do with a problem in generating hormones. It's a hormonal issue, whereas type 2 diabetes is simply a resistance to insulin, where you get so much insulin, you've been getting so much insulin throughout your life that you simply don't respond to it as well as you used to when you were younger. That's what type 2 diabetes is. Type 2 diabetes is an old people disease. For hemochromatosis, does not carry any of the hemochromatosis variants. For Alzheimer's, the APOE, by the way, this is the APOE gene is not what was on that Nebula result, right? If you look at just the APOE, he does not have any alleles associated with Alzheimer's. But yeah, so he's probably gonna have a very low score for Alzheimer's with my trait predictor. And of course, if he's scoring a low score for Alzheimer's with my trait predictor that features like 20 SNPs versus a 99 percentile score with Nebula, you probably want to trust Nebula over my trait predictor. As counterintuitive, as sabotaging as it is for me to say. So no micro P, you know what that is. I'm kind of afraid of even saying that because I recently posted a video and it got demonetized. And I'm thinking, I don't know exactly why, but I'm betting my money that it was because I mentioned this. So I don't want to mention it. You can pause the video and you can read what's on the screen, but I don't know. Maybe I'm already screwed because I already said it. So less likely to gain weight if taking Zyprexa. Here, more likely to gain weight if taking Zyprexa, and greater odds of cannabis-induced psychosis. Probably shouldn't smoke weed. I don't know if these ancient North Eurasians were smoking cannabis recreationally. Probably not. I do know that the Scythians and like the steppe people did. Scythians, Sarmatians, various Iranic people. Okay, for albinism type 2 doesn't have any, does not really have any albino mutations, and also is not a carrier of the Melanesian blonde hair variant in SIRP1. That's right, Melanesians actually have their very own variant in SIRP1, which causes a blonde phenotype for them. Nobody besides Melanesians has it. They did not get it from Europeans because Europeans don't have this blonde variant. Only Melanesians do. So let's see polygenic risk scores. For polygenic risk scores, he is scoring much less the average odds of schizophrenia for Northern Europeans. Does not have schizophrenia. I think for the Nebula result, it was like a 60th percentile, so this is much lower than what he scored with Nebula. For type 2 diabetes, he's actually scoring above average odds of type 2 diabetes, which compared with the Nebula result, which was 99th percentile, once again, is kind of really tame. And for Alzheimer's, but my risk score thing, it's using Bayesian methods. It's not using whatever Nebula is using to do their calculations. So maybe 1.7 times the average odds does translate into 99th percentile. Probably not though. Probably not. For Alzheimer's, this individual has much lower than the average odds of Alzheimer's with my calculation, which, as we've seen previously with the Nebula result is pretty much the opposite of that. He's scoring 99th percentile for Alzheimer's with Nebula. Well, that's pretty much all I wanted to show you here for the Malta boy. Leave your comments, what you think about the video. Leave that in the comments. You can download this file in 23andMe format from link which is in the description, and leave a like and subscribe because it was a lot of effort to do. It took me the whole day. My computer was not even just today, it was also a little bit of yesterday. I was downloading these files, I was mapping these files. It was a lot of work, guys, and I missed out on a lot of interesting stuff like watching Breaking Bad or whatever, but it did take a lot of time on my part, and I would appreciate it if you shared the video, liked, and subscribed. Goodbye.